So, hi guys. Today I'm going to talk about inheritance patterns, right? So we are looking at the way in which traits are being passed from a parent to its offspring. Right? And we're going to be focusing on organisms that reproduce sexually in this particular moment in time. I will look very briefly at asexual reproduction, but that's just so straightforward in terms of inheritance that it's not, something we have to delve deeply into. Right? And so most organisms are diploid, like us. I mean, they have two copies of each chromosome, one from each parent. Right? It's a very effective mechanism of creating genetic diversity within a population. Right? And so the gene pool doesn't have to gain new alleles to gain new combinations through sexual reproduction. Right? So that's one of the main benefits. That is the main benefit of sexual reproduction because it is very energy costly. Right? And so to spend all that energy to reproduce, there better be a big payoff, and that big payoff is diversity. Okay? And so when we are looking at inheritance patterns, we are talking about predicting the type of offspring that any two mating pairs might generate. Right? And so if I have a coyote boy and a coyote girl and they fall in coyote love and they make coyote babies, they make 50 coyote babies, how many of those 50 coyote babies are going to have gray fur versus brown fur? That type of thing is what we're looking at. Right? We want to be able to predict those numbers. Right? And so this all comes down to the way in which our genes are structured and the variations that we have in our genes. Right? And so in my little model here at the top, I have chromosome number 1 from some diploid organism. On this chromosome, we are looking at gene number 3 per the model. Right? Arbitrary choice on what gene number it is. But this gene 3 will always be found on this chromosome in this spot and will always code for the same trait. Sticking with the coyotes, this will be fur color. Okay? And so allele A is one variation in the coding of the DNA for this gene 3. Okay? So let's say allele A is the gray fur. Okay? Allele B is a different variation of the coding for that same gene. Okay? So something about the DNA sequence is changed between A and B. Okay, there was a mutation along the way, it's picked up different sequences, and therefore it's going to make a different ultimate protein and have a different trait expression. Okay? And so we go through the whole process of DNA transcription and then translation of the RNA into the protein. And because the alleles have different sequences, the proteins that are generated are going to have different shapes and ultimately going to have different functions. Right? They're always going to be for the same trait because they're the same gene, but they're going to have different presentations of that trait. Okay? And so alleles are technically the difference in the DNA sequence. Okay? You're going to have different alleles with the same phenotype. Okay? So the allele is not gray versus brown. These two alleles present in different ways, gray versus brown. Right? But the allele is the variation in DNA sequence. Right? So in sexually reproducing organisms, only half of the ploidy is passed from the parent to the child. Right? And so as diploid organisms, right, when I make a baby, okay, I give it one ploidy, haploid amount of information because I'm a diploid, right? So I have 2, I give my baby 1, my wife gave my baby 1, smush them together, the baby has 2, back to being diploid. And so this creates variations of the allele combinations that didn't exist in just me or just my wife, right? That baby now has a unique composition of allele combos, right? So there's that variation that the sexual reproduction is there for, right? So because we have 2 copies of every gene, right, we're ultimately making 2 copies or 2 variations of every protein for each trait. And these proteins are going to interact with one another in various ways. And that's what determines these kinds of patterns of inheritance, right? And so our typical or our traditional pattern of inheritance is what is known as Mendelian inheritance. Right? When we think of our freshman year Punnett squares, right, with our dominant trait and our recessive trait, okay, our big A and our little a, okay, that's Mendelian inheritance. Okay? For every trait, there's one gene. Each gene has two alleles, two different variations. Okay? One of those alleles is dominant, and one of those alleles is recessive. And so the dominant allele is the allele that will ultimately be expressed as an endgame, right, when you have a heterozygote organism. So if I have an organism that has a dominant allele and a recessive allele, you'll see the dominant allele in the expression. And I say see figuratively, right? A lot of these things are like enzyme rates. You don't see enzyme rates, right? But like on a data chart, you would. Okay? So the ultimate expression is the one that is dominant. Now, because of the rules of segregation of the chromosomes during meiosis, right? We talked about that independent assortment during our meiosis video. Right? We can predict how our allele combos are going to separate for each of the parents, and then how they might come back together for the babies, right? And we can use just raw probability to predict how many babies will get how many different assortments of alleles, right? Because it is random and therefore is governed by the laws of probability. So we have a genotype, which is just the actual assortment of alleles the organism has. Right? And so in my model here, I have a red allele and a blue allele. So I can have a homozygous red organism. It has two red alleles. I can have a homozygous blue organism. It has two blue alleles. Right? Or I can have a heterozygous organism. It has one red and one blue. It's a mix. Okay. So hetero means different. It's the mix. Homo means the same. Right? So it's the same, either red or the same blue. Right? I've started with two parents who are both, nope, they're not both. I thought maybe I made them both heterozygous because sometimes I do that, but this time I did not do that. I made the male heterozygous and I made the female homozygous red. And so we set up our Punnett Square. I'm sure you remember these bad boys, right? Now, I like the hashtag Punnett Square. I don't like the closed off Punnett Square. It's a waste of lines. Stop wasting your lines. Just make a hashtag. Boop, boop. Okay, and fill it out. So the male here, right, it started as a red and a blue heterozygous. Through the process of meiosis, those chromosomes, those homologous chromosomes, are separated into separate gametes, and one of those gametes is going to be involved in making these babies. Okay, and so this is a male, so it's a sperm, right? And so one sperm gets the red, one sperm gets the blue, and then it's random, right? They're both shot in there, right? And so whichever one makes it to the egg at the right time is the one that's going to determine whether the baby's getting the red version from the dad or the blue version from the dad, right? It's random. Okay, 50-50 shot, either one. From the mom, there was only red, right? So it's a 100% chance this baby's getting red from the mom. Okay? And then so we use this Punnett Square, which is a tool of probability prediction, okay, to figure out how many of our babies will have how many genotypes. Okay? So looking at the genotypes of the offspring, right, in this Punnett Square, I have two offspring that are going to be homozygous red. Okay? So two of the four are homozygous red. The other two are heterozygous. Okay? So this would say 2 of 4 and 2 of 4 because they have the same denominator. I can simplify them down to 1/2 each. Okay? So 1/2 will be homozygous red and 1/2 will be heterozygous for genotypes. Okay? Now, if we set the stipulation that red is dominant, okay, then for phenotypes, 100% of these babies will be looking red. Okay? That's it. Because the red's here, the red's here, the red's here, the red's here. They all got at least one red allele. They're all going to be red because that's what it means for red to be dominant. Right? Now, if we set blue as the dominant allele, right, our phenotypes will be evenly distributed. These top ones, will be red because they are homozygous recessive, right? And that's the only way to get a recessive trait to show up is to be homozygous. And these heterozygotes will present as blue. So it'd be 50% red, 50% blue for phenotype. Okay. And that's Mendelian genetics. Boom, done, right? We already know those things. That's from freshman year. Now, we should have also covered most of these freshman year, if not all of these freshman year, right? And so this should be mostly a refresher, but if not, we're going to talk about them and then you'll know. And it'll be a refresher next time we talk about it. Same thing, but different. Okay? So here are types of non-Mendelian inheritance patterns. So these are inheritance patterns that don't obey those rules of one trait, one gene has two alleles, one allele dominant, one allele recessive. If there's some variation in those rules, then it's a non-Mendelian trait. Okay? So, all right. We got multiple alleles. It's exactly what it sounds like, right? Instead of having two variations of this gene, we have more than two variations, right? So I have 3 variations, I have 4 variations, I got 5 variations. Okay? Codominance, okay, is when both variations, both alleles present together in the ultimate expression of the trait. Okay? Incomplete dominance is when both alleles present as a blend. Sex-linked is when the gene is on one of the sex chromosomes, right? Either the X or the Y. Okay, and that impacts the way in which the information is passed on to the child in a very unique way, right? Especially for us, right? So when I say for us, I mean organisms with 2 sexes, right? And that have different XY stuff. Yeah. We have polygenic, poly meaning many, genic referring to genes. And this is when one trait is coded for by more than one gene. Okay. And now most of our traits are actually this. That's actually how real genetics works. Very, very few traits are actually coded for by a single gene. So those are the ones that I'm going to talk about today. Then there's also epigenetic traits, which are traits whose expression depends upon environmental factors. And that topic will get its own lesson. Okay? Just like linked genes will get its own lesson. And linked genes are when we're looking at 2 or more genes and they're on the same chromosome. So the rules of independent assortment don't apply to them, right? Because they move together. And so that impacts the way that certain traits are passed along as well. And so that will get its own lesson as well. Okay, so today we're doing the ones that are not starred. Each of the starred ones get their own unique lessons because they are so cool and fun, and I love them. All right, so looking at multiple allele. Again, it's when there's more than 2 alleles for a single gene. Okay, when you're dealing with multiple alleles, you need to establish the dominance and recessive patterns, okay? Because it's not the same anymore. It's not just one dominant and one recessive, right? And so in this particular instance, I have 3 alleles. I have capital A, I have capital A prime, and I have lowercase a. Okay? And I've set the rules here that A is dominant to A prime, and both are dominant to the recessive A, the little lowercase a. Okay? And so I have a heterozygote male that has big A and little a, and I have a heterozygote female that has big A and big A prime. Okay? So I do my meiosis, I get my gametes, right? So this is a spermie, this is a spermie, this is an eggy, this is an eggy. And on this fateful day, if this spermie meets this eggy, the baby is going to be homozygous big A. Okay? If this spermie meets this eggy, right, I have a heterozygous big A prime. If this spermie meets this eggy, I have a big A, little a heterozygote. And if this spermie meets this eggy, I have a big A prime, little a heterozygote organism. Right? So it's 1 of 4 for all these different genotypes. Now, because I've set the rules that A is dominant to both the other ones, anyone that has a big A is going to be looking like a big A, right? And so this has a big A, this has a big A, this has a big A, this does not have a big A, right? So 3 of the 4 are going to be looking big A. The last one is going to be looking big A prime, right? Because big A prime is dominant to little a. Okay, so that's how multiple alleles work. You just throw in more alleles than the usual amount. Yeah, they can have different dominant recessive relationships as well. Like it doesn't have to be sequential like this. Like big A could be dominant to big A prime, could be dominant to little a, but little a might be little dominant to big A, right? But all the rules have to be established for multiple alleles. There's no like base rule like there is the big A is dominant, the little a is recessive in Mendelian, right? We don't have those rules. Codominance. Again, this is when the expression of the gene demonstrates aspects of both alleles. They're showing up together, together, like co-workers. Okay? And so in this model, I have big A being blue, and I have big A prime being red. Okay? So a homozygote organism that is big A homozygote, this homozygote organism, is going to be looking blue. A homozygous organism that is big A prime is going to be looking red. Okay? But if I have a heterozygote organism in this particular setup, that organism is going to be blue and red. Okay, so maybe it's blue with red stripes, or it might be red with blue polka dots. Okay, you're going to see blue, you're going to see red. Okay, codominance, they show up together. You see them both. Okay. Incomplete dominance, very similar idea. Okay? Slightly different. The expression of this gene demonstrates a blend of both the allele qualities. And so if we stick with the colors, right, homozygous big A is going to be blue, homozygous big A prime is going to be red, and a heterozygous organism is going to be purple. It's like if I blended blue and red together, I get purple. Right? In case you didn't know colors, if you take blue, you take red, and you mix them up, you get purple. It's a blend. Okay. This also works with other traits. Like this isn't all just color stuff, right? This isn't all just stuff you can see. Right? So maybe this is enzyme rate, right? And so big A homozygous is fast. Big A prime homozygous is slow, okay? And a heterozygote organism is going to be a medium rate. If I blend fast with slow, it's medium. Okay? Incomplete, blendy blend. And then we get on to sex-linked, which is kind of cool in that it changes like the actual way that inheritance shows up. Okay? And so this is when the gene is on one of the allosomal sex chromosomes. Right? And so allosomal means that the chromosomes are different from one another, as opposed to our other 22 chromosomes, which are autosomal. So autosomal means that they're the same, right? Like number 1, it looks like number 1. Number 2 looks like number 2. Number 3 looks like number 3. But you get to number 23, right? X does not look like Y. They're allosomal. Okay? So if it's X-linked, the gene is on the X chromosome. Mind-blowing, right? If it's Y-linked, the gene is on the Y chromosome. Somebody did a good job naming these one day. Okay? Now, males are always hemizygous for any sex-linked trait. Hemi means half, right? Like a hemisphere. So they have hemizygosity for any sex-linked trait because they have one Y and one X. Right? So if it's X-linked, they got one copy. If it's Y-linked, they got one copy. Females never have any Ys. So they are not anything. There's no description for them. They're not Y-linked. Okay? They are always going to be properly zygotic for X-linked. Right? They'll have 2 copies of their X. Now, an interesting thing is that you do, as a female, I don't do it because I'm not a female, but if you're a female, right, in any given cell, one of your X's is turned off. Right? So you only have the presentation of one X in your cell. And it's random. Right? So through the wholeness of your body, you get all of them, but in any given cell, you got one X turned on and one X turned off. It's called a bar body when you get the one turned off. And we might talk about how that happens. It has to do with cell communication, so it might tie back to some of the stuff we talked to in the past, which is always nice. So we might review that. All right. So we're looking here at the top. We got a female. It's XX. Genotype-wise, she's X prime X. And my male, I made him X prime Y, right? So males always be XY, females always XX. Okay? Now, when they're making babies, right, if this spermie with the X in it meets this eggy with an X prime in it, that baby right here is going to be girl. Well, it's going to be female, right? Girl's not a real thing. Girl's an imaginary social status, but female is a real biological thing, right? So this is female, right? And this female is X prime, homozygous. Okay, boom, that's her. So she's going to be looking X prime, period. Right? I set X prime as dominant, so anyone with X prime is going to be looking X prime. If this spermie meets this eggy, this female is going to be heterozygous because X prime is dominant. She's going to be looking X prime too. Okay? If this spermie meets this eggy, it's going to be a male, okay? And it's going to be looking X prime because it has an X prime. If this spermie meets this eggy, right, it's going to be a male and it's going to be looking X. Okay? So the phenotype representation is all the females will be X prime in this particular instance because they all have at least one X prime. Whereas the whereas the males will be evenly split between X prime presentation and X presentation. And so that's the way in which sex-linked traits change the inheritance patterns, right? Males and females will have different rates of expressions of phenotypes due to the fact that the male is hemizygous, right? Whatever X the male gets from the mom, that's what X he's going to present. He doesn't have another X to cancel it out. Okay? And so for recessive traits, this is particularly important because there's nothing to cancel out a recessive trait. Right? And so most of the time you get recessive trait canceled by a dominant trait. In this case, you don't have that option. Okay? And whatever information he gets from his dad on his Y is going to be what he has for a Y because the mom can't give any Y information. Right? And so males are more strongly impacted by sex-linked traits than females are due to this homozygosity aspect of it. Okay. And onwards and upwards to polygenic traits. So I'm just going to describe a couple here. I didn't draw them out. Okay. TBH, because my whiteboard was starting to lag. So it was getting annoying to draw. So I'm going to draw them with my mouth. Okay. So polygenic traits are when traits are coded for by more than one gene. Right. So skin color is a very traditional example in humans of a polygenic trait. And so skin color is determined almost entirely by the amount of melanin pigment that we have. And so when you think of it being only one protein, the question would be, if it's only one protein, how can we have such a myriad of colorations in our human race, right? So we have some humans who are really, really, really strongly melanated, super dark, right? And then we have some humans who have no melanin, they're broken, right? They're albino, they can't make any melanin, super pale, right? And then there's anywhere in between, right? So if it had gone by any of the things that we just described before, you'd really only have three potential options. You'd have super pale, you'd have super dark, or you'd have somewhere in between, right? So we'd have homozygous recessive, homozygous dominant, and then heterozygous. Okay? But obviously that's not how that works because we don't just have three skin tones, right? We have this whole like scale of skin tones anywhere in between. Right? Now, skin tone is an example of a summative trait. Okay? I have a bunch of different copies of that melanin producing gene. And each copy obeys simple Mendelian inheritance rules. Right? And so if I have recessive on chromosome 1, I have dominant recessive on chromosome 2, I have recessive on chromosome 3, I have dominant on chromosome 3, I have recessive on chromosome 3, right? All those are going to do their thing individually. Okay? And so if I have two recessives, that's not going to make melanin on whiteness, right? And then I got one over here, I had a heterozygous, so I can make some melanin. So now I got a little bit of pigment, right? And then up here I had dominant. So then I got two more melanins, good. So I got more melanins walking around, right? And then I had another recessive, no melanin, so I'm still paleness, right? And then I had another heterozygous, so I have another melanin, right? And so each of those alleles that are capable of producing melanin is adding more melanin to my complexion, right? And so the more alleles that I have that work, the more melanin I'm going to have. Okay. So that's how polygenic traits work in a summative way. Right? Each individual gene is still expressed away from the other ones, right? They're not interacting with one another. At the end, the proteins kind of like build up as a totality of how much of that protein you have. The more interesting form of polygenic traits are epistatic traits. Okay? And so traits that are regulated through epistasis. Now, this is when I have a gene that codes for a specific trait. Let's say it's hot dog enzyme B. Okay, and so hot dog enzyme B is what determines how fast I can break down hot dogs. Okay, and so if I have the dominant version, I break down hot dogs really fast. If I'm homozygous recessive, I break down hot dogs really slow. Right, if heterozygous, it's somewhere in between. Okay, so that's my hot dog breakdown gene. Okay, and it's just floating around breaking down hot dogs. Okay. Over here, I have my epistatic gene. Okay, it doesn't break down hot dogs. It's not its job to break down hot dogs, but it's going to impact this hot dog breaking down trait over here. Okay, it's going to make a protein. Okay, that somehow impacts this enzyme's ability to break down hot dogs. Okay, and so maybe this protein attaches to my hot dog breakdown enzyme and makes it go really fast. Right? Could do the opposite too. It could attach to the enzyme and make the hot dog protein not work. Maybe I can't break down hot dogs. Okay, and so this epistatic gene is going to have its own allele set, right? So I'll have dominant is produced, recessive it's not produced, right? And so this epistatic gene impacts my original enzyme gene. Right? And so that's what this epistatic relationship is. Okay? It's when non-allele genes can impact other genes. Okay? So like it's not their own alleles interacting. It's not one version of an enzyme interacting with another version of the same enzyme. It's an enzyme getting impacted by a different protein from a different gene to regulate its expression. This protein could also glob onto the gene that made the enzyme in the first place and just completely cancel out. That's another way that epistasis can work. Okay, but the idea is that I have this side gene impacting the expression of my original gene. Yeah, that's it. I figured it was going to take me a while to get through those, and it did. But good, it's worth it. I love these things. These things are one of my most favorite topics. I actually like genetic regulation better, but these are like right there with it, like gene regulation in this are so intimately intertwined that they're basically the same topic, which is why we're doing them in the same unit, right? So this is still about gene expression. This is just about how gene expression shows up in organisms as actual traits and how it gets passed on to babies. That's it. Have a great day. Bye.